بيور epidermal cells تعتبر epithelial cells الموجودة في الطبقة العلوية الجلدية and they fall apart from the least amount of force that it experiences. Okay, so the cell function helps it with that. And any defect in these protein complexes, any mutation can lead to severe diseases. مثلا عندنا in a skin, for example, okay, if you lack certain kind of junctions that hold the cells, the epidermal cells together, okay, or the epidermis to the underlying matrix, you will have like very severe and could be fatal skin diseases, okay, and other kinds of diseases as well. So you can have cardiac disease, you can have deafness, you can have all sorts of things depending go wrong depending on the kind of the junction that's being affected. Okay. So the way we classify the junctions, they are by location. Either مثل ما قلنا located between two cells, hold two cells together, or hold the cell to the matrix. فعلى الأساس we divide them to cell to cell and cell to matrix. So this is by location. There is another kind of classification by function. Okay. So if we look here, we will see that we have, based on the function, three kinds of junctions: anchoring junctions, occluding junctions, and channel forming junctions. So an anchoring junction, from the name, they anchor or they attach in a tool. Okay, so they attach two cells together or the cell to the matrix. These are the only kind of junctions that you can find in these two locations. The other junctions, the occluding junctions and the channel forming junction, you can find them only between two cells. Okay? And from the name, the occluding junctions, they occlude. صح? Or they impede. يسدون. فإذا you will see that they are located here, like in the apical border on top. And what they do is that they control or occlude the movement of molecules to from one compartment, this epithelial, the extracellular environment here, near the apical surface, and the extracellular or interstitial fluid here, near the apical lateral surface. So, and we will talk about it later, like there will be some movement, but it depends. Certain ions or certain molecules can pass through. The channel forming junction, راح نشوف إن هم intercellular channels that immediately attach the cytoplasms or connect the cytoplasm of two cells directly together. Okay? فعشان كده نسميهم channel forming junction. And they allow communication between two cells directly. Okay? Alright, this is artificial. We will have all of these in a classic situation, the epithelial cell. Okay, we will have all of these together. And they are arranged in this format. So basically you have the tight junctions up top, but then you have the adherent junction, anchoring junction. So two kinds of anchoring junction. Okay? راح نتكلم عنهم الadherent junctions and then the desmy zones and then تحت راح نشوف الباب junctions. Okay? طبعا here we have two kinds of circulators anchoring junctions. اللي هما الهيدرين زون and the actinide cellular junction كلهم راح نتكلم عنهم. Alright. مثل ما قلنا tight junctions, they are found in the apical border of cells and they seal this gap in intercellular space. Alright. If we zoom in, راح نشوف إن هم شنو؟ It's a network of strands. مثل الحبال مشكلين شبكة. Okay? A branching network of strands. طبعا on both sides. يعني هني هذا lateral membrane of cell 2 وهذا lateral membrane of cell 1. إذا هذا strand الطرف المقابل في strand similar strand. Okay? And they interact together مثل السحاب. Okay? What they do is they just they seal the space where they interact. Alright? And the places where you don't have that strand راح تشوفين إنه في شوية space. Okay? branching. The more branching you have, the tighter the seal, okay? There is also another protein called occludin. The occludin, it's not important structurally. If you don't have it, the tight junction will still work. But it will be more permeable or more leaky compared to when you have the occludin. It adds another layer of tightness, okay? So these basically, this is, if you look from the top, you will see a portion of the stand, the ceiling stand, coming from both sides, stand 1, cell 1, and cell 2. We will notice, in claudids and occludins, we will see that the interaction between these proteins of the ceiling stand is homophilic. Homo, same, philic, like or love. So an occludin from one cell interacts with occludin from the claudin from one cell, interacts with the claudin of the other cell. The extracellular domains interact. Pretty much, I think there are 4 transmembrane proteins, and their extracellular domains interact together from the opposing cells. The occludin interacts with the occludin from the opposite cell. Okay? So that's why they are homophilic. Alright, we already said that. Okay. There is another kind of protein that provides the tight junction. In claudids and occludins, you will find them where? The bicellular contact. When the two cells next to each other, you will find them. But they will form little down. Okay? Horizontally, up top of the cell. Imagine if you have a tricellular contact, 3 cell walls coming together, هالشكل هذا. Okay? راح يصير gravity. ممكن يصير leakage. So to tighten or to seal this space, okay, يسمونها central tube, you have another kind of protein in the gap junction that runs vertically. يسمونها the tricellular. Okay? So the tricellular, again, if you look up top, راح نشوف ان الترايسيلولر راح يشكل, you will have one from each cell, راح يشكل little central sealing element. This structure will seal the central tube and will limit the permeability. Okay? So this is all revision, صح? So هنتكلم عن more about the tight junction. So بالنسبة للfunction, and there are 2 main functions of the tight junction. They can act as a fence and they can act as a gate. So this is the easiest way to remember. Okay? What do I mean by a gate and what do I mean by a fence? So a gate, طبعا, it's guarded. It doesn't open to anyone. صح? Put in a security guard or whatever, like a scan card or something to have access. Same thing here. So if you have like these cells interacting with like tight junction, you will see that basically it will act as a gate, meaning anything in this compartment here, in this extracellular area here, will not pass down, and anything in this basolateral extracellular fluid will not come in. Okay? imagine you have a garden or a yard in front of your house, and your neighbor has one in front of his house. And the neighbor kids play basketball or football or whatever, and because there is no fence that separates your yard from your neighbors, they can just forget, and then they just can come into your lawn and play there, okay? Well, your kids can go the other way around. They can play and they end up in front of your neighbor. Because in the image fence, you will make sure that the kids stay and play in your yard, on your lawn, okay? So نفس الفكرة هي tangential function as fs. Because why do I say that? لأن إحنا عندنا الapical domain والbasolateral domain. البروتينات, الcarriers, channels, you have also certain kinds of lipids here, okay? They can move, okay? It's not like there's, إذا موجودين they can move laterally. Imagine they move laterally and there's no tight junction. They will end up here. So راح يكون من الapical domain to the basolateral domain. And the ones in the basolateral domain راح يتحركون laterally. So ممكن يصيرون إذا ما في tight junction they can go up and end up in the apical domain. فراح يصير mix. طبعا we don't want that. احنا نقول الانثيليال cells are polarized. شو المقصود بالpolarized؟ مو opposite. شو يعني opposite؟ شو المقصود؟ I'm sorry. They are not excitatory. لا لا لا. Polarized المقصود فيها they have two poles. Okay, عندهم apical domain و basolateral and they are not the same من ناحية structure و function. مثل ما قلنا في certain carriers موجودين or transporters في apical surface. في others موجودين في basolateral surface. And they stay where they are. Okay, they stay where they are to make sure that the epithelial tissue function optimally. Okay, وراح ناخذ example. طبعا this is important for the transcellular transport. Okay, gate function مسؤولة عن شنو؟ regulating the paracellular transport. بس الtrans function مسؤولة عن الtranscellular. Transcellular اللي هو the transport across the destroying what I'm doing. What's the point? So, section 3 type junctions are not permeable head disease. So that's why like you see how a tight junction, they keep everything where it is so that they function in coordination and عشان يكون optimal function of this intestinal epithelium. Okay? طبق هذا على different kinds of epithelium. Okay? So we said that. في سؤال so far? They allow, they can. Some leak more than a little bit of water. They allow, depending on the clotting in the water, they can allow sodium to pass or chloride or whatever, depending on the junction. Some junctions are tighter than the others. يعني مثلا عندك tight junction موجودين في intestinal epithelium. They are many times, thousands of times more permeable than the tight junctions that are moving the epithelial cells together. من ناحية الصوديوم. Okay? So the sodium drives water and so they are more permeable to water compared to the tight junction or bladder. Okay? So it depends. Now let's talk about the communicating junctions and the ulna. Another name for it is like the gap junction. So it's basically an array of intercellular channels, and they directly act in channels that connect the inside of the cell, the cytosol, to the outside world, to the extracellular environment. These connect two cells together. The cytoplasm is two cells together. So cytoplasm is the membrane of cell 1 and this is 2. And they have these half channels or hemi channels. Each one gives a hemi channel and then they stick together and they form a whole channel. طبعا هذه channel can allow certain molecules to pass. They are again water filled, okay? They're hydrophilic, so they allow, because they're water filled channel, they allow polar molecules, but they have to be very small, okay? Anything less than 1 kilodalton polar can pass. Okay? So some ions can pass, and like some metabolites, small metabolites like from metabolism, we have ATP, nucleotide, cyclic AMP, some references saying glucose, but I'm not so sure about that. But anything that is polar and less than 1 kilodalton can pass. Okay? Second messengers, calcium, inositol triphosphate. In macromolecules, they're excluded because from the name macro molecules are huge. So proteins or polysaccharides, okay, not like small sugars, they cannot pass. Okay, or nucleic acid, RNA, DNA, they cannot pass. So they are expressed in. The diameter is 1.5 nanometers. So anything bigger than 1.5 nanometers or close to 1.5 will not pass through. Okay? And you will find it in most tissues. Okay? I can show you in the heart tissue, I can show you in the epithelial tissue, I can show you in the neurons. You'll see them everywhere. Smooth muscle. Alright, each gap junction is عبارة عن two channels or two hemi channels. يسمونهم connexon. So this is a connexon and this is a connexon. This is a connexon. And the connexons or the hemi channel عبارة عن شنو? 6 monomers. Okay? And each monomer is called a connexin. So we have a connexon, which is a hemi channel or half a channel, 2 connexons, or a gap junction, one gap junction channel, or a gap junction with multiple intercellular channels. And then each has, each connexon is made of like 6 monomers that go from in connexins. And so if you have a connexon or a half hemi channel that all the monomers are the same, so we call it homomeric. Homo, same, meric, monomer. All the monomers are the same. But if they have different kinds of monomers, they have meric. And depending on the kind of connexon, like if you have the first connexon similar to the second connexon, potential very quickly, so the cardiac muscle can pump, get activated at almost instantly at the same time, and contract as a sensation. Okay? So they are very important. راح تشوفون موجودين في الانتركرياتيف discs. So they allow very fast and rapid depolarization and activation, which of the excitable cell is cases like the heart muscle, which allows synchronized contraction. Same thing that applies to smooth muscle cells in the intestine and the uterus and the bladder. Now, also you find them in other kinds of excitable tissue in the nervous system. Okay, they form what is referred to as electrical synapses. Okay, and after chemical synapses, right? Here we have electrical synapses. Because we have gap junction, they also allow the electrical current to pass directly from the cytoplasmic mucosa to the. Okay, so this is actually very important when you have a large group of neurons, interconnected neurons, and you want them, of course, to fire synchronously at the same time. Okay? It allows very rapid transmission. Okay? عكس الديلي الموجود في الكيميكال synapse. صح؟ لأن it still needs to release the neurotransmitter, it binds to the receptor in the postsynaptic membrane, and blah blah blah. This one is less than, make sure electrical current is conducted with less than 0.1 milliseconds. So it's very rapid, and it's also bidirectional. The synaptic transmission, you go from one direction to the other, from the presynaptic cell to the postsynaptic cell. And you can go both directions. And having bidirectionality is important, especially when you have a large group of interconnected neurons working together or firing together. Okay? It helps them synchronize their action, and it also makes the neurons more sensitive. So this is just again a vision. Synaptic and electrical synapse compared to a chemical synapse. Okay? طبعا non-excitable cells also have the gap junctions, and they share مثل ما قلنا like ions, sodium, chloride, metabolites, ATP, ITC, second messengers, and so this way they are metabolically coupled together. they do, they link the outside to the inside. Inside the cell, the cadherins are bound to the cytoskeletal filaments, either actin filament or intermediate filament, depending on the kind of the anchoring junction, cell-to-cell anchoring junction. بس طبعا البايندينج of the cadherin to the cytoskeleton, it's not direct, it's indirect, لأن في adaptive proteins تساعدهم in that sense, okay? So بالنسبة لي عفوا the cell to matrix anchoring junction, الانتجrin, it binds outside the cell, it doesn't bind to another integrin. Again, cadherin, killing transmembrane protein or transmembrane adhesion protein bind to something outside or inside. Okay, cadherin binds to a similar cadherin on the outside. Okay, the cell integrin to the outside of the cell binds to an extracellular matrix, a protein that belongs to the extracellular matrix. And on the inside, it's anchored to the again cytoskeletal filaments through adaptive proteins. فهني راح نشوفين كأنه continuation, يعني كأنه الcytoskeleton of the cell, this cell, in a way, it's indirectly continuous with the cytoskeleton of this cell, لأنه عن طريق الcadherins. So, in a way, they kind of coordinate like their contractile function. That the actin is contracting on one side, this cell also it will contract if it wants to balance the force, because we don't want force in one cell pulling on one cell and like more than the other, so that we don't deform the tissue. So this is the point of the junctions. Alright, and also this maybe integrins of the cell to matrix anchoring junctions, they in a way link the outside of the cell and link the extracellular matrix outside to the inside of the cell in the cytoskeletal filament, so they can communicate as well and transmit forces. Okay? هنتكلم عن الادهيرنس junction. So these are cell-to-cell junctions. And they are very common in epithelial cells. وقلنا they have the terms membrane adhesion protein belongs to the superfamily cadherin. بس في حالة الادهيرنس junction البروتين هي اسمها classical belongs to classical cadherins. And the classical cadherins they're calcium dependent. So normally they're floppy, okay, but when the calcium binds, we have two membranes and we have two classical cadherins. By the way, they are the same. Okay, they're the same, they're just in different colors, the exact same thing, different colors, just to distinguish cell 1 from cell 2. So we see that the N-terminal tip of the extracellular domain interacts together. ليش? Because of the presence of calcium. إذا الكالسيوم ما كان موجود, then here we have 1, 2, 3, 4, 5 repeats, cadherin repeats, and بينهم hinges, flexible hinges. إذا ما كان الكالسيوم موجود, it's going to be floppy. But if the calcium binds, they will become like stiff, slightly curved, and it will enable the interaction of the internal tips together. Okay? So these are the classical cadherins, and they are homophilic. So that's why I emphasize this one is the exact same thing as this one. And the cadherin binds to a similar cadherin on the exact same kind of cadherin on the nearby cell. Okay? وطبعا قلنا ان inside the cell, this is inside the cell is not shown, but the adapter proteins indirectly links into the cytoskeleton. But which cytoskeleton? The actin filaments. Okay? So this binding is actually low affinity. Okay? Because if you don't have one, you have an array of them, you have tons of them. And so the more you have multiple low affinity interactions, the overall effect is very strong anchoring and attachment. Okay? So if we look at the cell when you look at the microscope, okay, the cell under the الadherence function. Okay? And if you need more information, you can in the class from here, you can go to this link. Alright. طبعا الفانكشن مالت الadherence function من الاسم. Integrity, exactly. To give the cell integrity. And hold the cell together, give it mechanical strength to resist any pulling or any forces on it. But that's not the only thing. Okay? It's also important in embryonic development. لأن these structures are not static, or they look like they are static, but they are dynamic. Okay? And they can play a role in migration. So let's see how this happens. خلينا نطبق على الانتجريتي. خاصة في التشو اللي معرض حق a lot of mechanical forces within the heart. صح الهارت لأن it pumps all the time. So you want the cells to kind of be held together. مثلا image skin, okay? The blood vessels لأن على طول في blood moving في friction. So we want the cells to stay held together. Now, شلون يلعب دور? How do these adherent junctions play a role in embryonic development or embryogenesis? أكو خاصية في الكاديرنز, and we know that cadherins bind homophility only to the exact same type of cadherin. So this will help the عملية اللي هي tissue separation. شنو أقصد بالtissue separation؟ يعني لما الامbryo it develops, it goes from something simple to something more complex. So, like initially you have one germ layer, but then يصير عندك 3 germ layers, ectoderm, endoderm, mesoderm. You go from, we will take this example, you go from a neural tube, like a primitive nervous system, to spinal cord, brain, and then like peripheral nerves. This is a very dynamic process, and you need basically the cadherins play very important role in this process. So الخاصية هذه اللي تساعد على tissue separation and embryogenesis, خاصية الكاديرنز يسمونها sorting out. What do I mean by sorting out? So if in the lab, in a culture dish, you grow 2 kinds of cells, okay? The red and the blue, okay? You tag them with a fluorescent dye, so they appear under the microscope in different colors. These cells express different cadherins. So the blue express a kind of cadherin or isoform of cadherin called E-cadherin. And E-cadherin is very prominent in the epithelial cells. The red ones express N-cadherin. And N-cadherin موجود مثلا في nervous tissue, muscle cells, okay? So this is, let's say we're doing this in the lab. And we let the cells grow. We will see that the cells actually will clump, they will aggregate. The ones with the N-cadherins will cling together, and the ones with the E-cadherins will cling together. Okay? So this is the homophilic binding. So they will sort out. هذه العملية تساعد في عملية tissue separation, في عملية embryogenesis. Not only that, but even if you have 2 populations of cells in a dish, they both express E-cadherin مثلا, the same kind of cadherin, but different levels. واحدة high level, واحدة low level. They will see that the blue ones that express the high level of E-cadherin, they will cling together strongly, and they will localize to the floor. They will form like a ball, and then the ones with the high E-cadherin, level E-cadherin will end up inside. Okay, and the ones with the low cadherin will end up outside. So تخيلوا لو هذا يصير بالembryogenesis يساعد على التشكيل، صح؟ So هذه الخاصية, this property of sorting out of the E-cadherin, which comes from its homophilic tendency, allows for this. Okay? So let's take the example of the formation of the nervous system. Okay? Let's say in the vertebrate or human. This is very simple. This you have each layer will give rise to different organs. And we can علشان يطلع لي another tissue. Some of the cells here will lose the A cadherin and it will express a different kind of cadherin with one N cadherin. فايش راح يصير؟ These cells now اللي هم كانوا أساساً نفس التشو. Now this tissue separated into the ectoderm separated into new tissue just by turning off the A cadherin and turning on the N cadherin. And now we are forming a central tube, اللي هو the primitive spinal cord or the primitive central nervous system. Okay? طبعاً it will invaginate until it separates. فراح يصير عندنا طبقة ectoderm وراح يصير عندنا neural tube. Okay? Now, some في سؤال لي؟ هذه example this is just to explain how the cadherins like to interact together. Okay? In the lab بس طبعا نطبق الخاصية دي خاصية sorting out هي يعني هي مثلا عندنا you will say that cadherin one kind of cadherin صح؟ Now if some of the cells they lost the A cadherin and express N cadherin راح يصير different kind of tissue and they will separate. شوف they will separate. راح يصيروا two tissue لأن they don't like each other. Spatially will separate. فراح يصير عندنا هي ectoderm and وكانوا هما نفس التissue. Alright? Now again, some of the cells in the neural tube, okay, or in the neural tube that will form the central nervous system. Some of the cells they will lose N cadherin. Right, they will gain another kind of cadherin and they will cadherin 7 okay? راح يصيروا different kind of cells. راح يصيروا neural crest cells. And they will separate again. And they are expressing different kind of cadherins. They just do sort out. They separate. So we will see the smooth aggregate of cells. Okay? These are the neural crest cells. Neural crest cells will separate from the neural tube and they will migrate. Okay? They're easier to move. They're loosely attached, they will migrate to other locations, and then they will form the ganglia, the peripheral nervous system, okay? Shown by turning off the cadherin 7 and again turning on an N cadherin. راح يصيرون neural tissue, but now somewhere else. فهي هذه الفكرة. These cadherins don't like each other, they just like themselves. And whenever you have another kind, they just separate from each other. So عشان كذي embryonic development تسبب خاصية الكاديرين تلعب رول كبير. Okay? Okay, so خلصنا الadhesion junction. أول نوع من cell to cell adhesion, cell to cell actin junction. The other kind of cell to cell actin junction is the desmosome. Desmosome similar to cadherin, نفس الشيء. عندهم transmembrane adhesion protein belong to the cadherin family, but the difference in it's not classical cadherin, it's non-classical cadherin. Or desmosomal cadherin. That's different kind of cadherin, okay? And the linkage to the inside of the cell, to the cytoskeleton filament, is not to actin to cell adherence junction, but to intermediate filaments. With intermediate filaments, very strong compared to actin. Actin is more dynamic. Intermediate filaments are rope-like, so they have high tensile strength. So when you just stretch them and pull them, you apply force on them, they don't break easily. Okay? فبما أن الدesmosomes are anchored or use our attachment site for the intermediate filament, we will expect the desmosomes to become very strong kinds of junction. مقارنة بالadherent junction. أقوى. Okay? So, again, this is the structure. Okay? And then the desmosomal cadherins or non-classical cadherins attached to interacting from both cells. To the inside, they are linked to the intermediate filaments indirectly عن طريق adaptor protein. لكن adaptor protein هذه الطبقة الحمراء هذه it's just and you can see it under the transmission electron microscope. It's very visible. The hectile intermediate filaments, you will see it's very visible under the microscope. The main function of the desmosomes, again, mechanical integrity. Best, they are even stronger than the education, and they are not as dynamic. Okay? لأن they are anchored to intermediate filament, not to active filaments. Okay? So they are very common in tissue within heart tissue with skin. Okay? لأن they are under a lot of mechanical stress. طبعا الانترميديate filaments اللي عندنا can be either keratin or desmin. The keratin, if this desmosome was found in the epithelial cell, then the intermediate filament is keratin. If the desmosome is found in a heart muscle tissue, the intermediate filament is desmin. Okay? في سؤال؟ anyone appreciate me? في سؤال؟ Also, there is non-classical patterns of desmosomal patterns. The structure, their structure is much more ordered compared to a DNA. So under a microscope, the desmosomes, you will find them as spots, very dense spots. And has two cells attached by desmosome. ما راح تشوفينهم continuous belt nerves in the adherent junction, and continuous spots. And when you zoom in on it, have a plaque, and this is one desmosome. So they hold the cells together tightly, have a plaque of adaptive proteins, and these are the intermediate filaments on both sides. Okay? الحين let's move to the cytomatrix junction. عشان هذا كله أكثر حفظ يعني. السنتوميتريكس junction قلنا عندنا شنو؟ نوعين. أول نوع اللي اسمه كمان الأكتين لينك سنتوميتريكس junction أو باختصار الفوكال. والنوع الثاني اللي هم الأساسي. So أول نوع راح نتكلم عنه اللي هو الفوكال adhesion. الفوكال adhesion structure, again, transmembrane protein. This time it's an integrin. Okay? An integrin is a heterodimer, right? So it attaches the extracellular matrix protein موجودة outside the cell to the actin filaments inside the cell عن طريق adaptive proteins. So لو تلاحظون هنا طبعا الانتجرين it's a heterodimer, it has alpha subunit and a beta subunit. Okay? And the tail of the beta subunit interacts with the adapter proteins. And then a major adapter protein we have talon. And then another one that assists the talon in its function in the operculum. The talon is the main adapter protein. So كأنه scaffold, okay? ينبني عليها or you attach to it multiple actin filaments. So you have it unfolds and then you have multiple kinds, you have multiple vinculum proteins, another kind of adapter protein. Yeah, يعني talon either binds to the actin filament directly or عن طريق الvinculum. Okay? So this way, the more it unfolds, the more vinculum binds to it, the more actin binds to it, and the stronger the adhesion becomes. Okay? طبعا الأكسترا كانت الانتجرين to the outside of the cell, it interacts or it binds, the head of the actin binds to the collagen or laminin or fibronectin. Okay? With focal adhesion. So here راح نشوفهم كأنه in a way an ECM is talking to the cytoskeleton and the cytoskeleton is talking to the ECM. Okay? A lot of communication going on. A lot of cell signaling. There are other signaling proteins here that we're not going to focal adhesion kinase, but I'm not going to talk about them. So collagen again, integrity, attachment of the tissue to the extracellular matrix عشان التissue becomes a tissue. Okay? بس احنا بدنا they are dynamic and they also play a role in cell migration. Okay? لأن imagine a cell احنا عندنا cell movement. مثلا let's say you have tissue injury. اللي ورا في سؤال؟ بنات في سؤال؟ Okay. If you have a question, please raise your hand. Stop me. Imagine you have a tissue injury, okay? You have a wound, you have a cut. So you injure many layers, صح? You injure the epidermis, you bleed because like you cut the capillaries. So how do you repair the tissue? You need some cells, definitely in the epidermis, you need stem cells to migrate in order to replace the dead or the injured epidermal cells, صح? You need fibroblasts to migrate from the connective tissue, connective tissue, rich in fibroblasts. They will migrate, they will move, and they will release collagen, okay, and elastin and all of these things. So you need movement. So how do cells move? They have to break the attachment. Like attached to some cells attached to extracellular matrix. They need to lose their attachment at the nearby cells and lose their attachment to the underlying matrix. And then they form it. So, let's take the example of a fibroblast are single cells. They are not tissue anymore. They use the extracellular matrix. A fibroblast wants to move. But it doesn't move with varroa. It's stuck to an extracellular matrix. So it has, like, in the anterior portion of this is called the leading edge. So what's going to happen is that it's going to extend, attached at multiple sites of a focal adhesion to the matrix. Now, if I want to move, an anterior portion of the leading edge has to break one of the focal adhesions, or a few of the focal adhesions in the front, and extend, and then make a new attachment. The body of the cell, it will be pushed forward. And a rear end will break a focal adhesion. It will lose attachment, and then make a new one to locality. So, chrome, and then, so, No, this is just an introduction and movement. And you said it's crawling. And I will send you a video of how the cell crawls. You don't need all of it and all the normal proteins and CDC42 and whatever. You don't need that. Just until minute 3 or so, 3:13 or so, you will see if it's helpful. You will see how the cell moves, how you become a single cell. But then also, a whole tissue can also move, but this also is too complicated. Just to get the idea, in the attachment, the making and breaking of the focal adhesion helps the cell move, and you need the cell movement مثل ما قلنا في الwound healing, في الembryogenesis, صح؟ هذا يسمونه الابيثيليال to mesenchymal transition إذا سمعنا. Physiologically it happens, okay? But pathologically it also happens. شلون لما يكون عندك epithelial cells, okay? لما يكون عندك epithelial cell that like a tissue, some of the cells break their attachment and they just leave the tissue and they migrate somewhere else and metastasize. This is how metastasis happens. Okay? طبعا آخر شيء الهيميديزمزوم, I'm just gonna go quickly. Hemidesmosomes, we have also integrins, that's a specific kind. You have alpha-6 beta-4, and you have collagen type 17. And mو بس الانتجرين, you have transmembrane protein. You have the integrin, alpha-6 beta-4, and collagen 16, and then a certain adapter protein. not direct